My name is Chris Sermo. I'm the Dean of the College of Letters and Science, and it's my pleasure to welcome you to the Community Lecture Series. What we try to do with these lectures is bring our expertise and our scholars and our teachers into the community so they can talk a little bit about what they do with the other 75 hours a week that they spend working. Having said all of that, our last speaker of the year will be on May 10th at 7 o'clock right in this room, and that will be Jody Lewis of our psychology department. She'll be talking about social class in education, psychological factors limit upward social mobility. Jody's an engaging speaker, and I think you'll very much enjoy her talk. We're here tonight to celebrate the chemistry department a little bit. They usually populate our lecture series quite well. There's usually one or two chemists here at every single lecture, particularly Lori. She's here at pretty much every one. Having said that, our speaker tonight has a really interesting background. That's all I'm going to say. And you can elaborate on this if you'd like. I'm going to try a little bit to talk about you, but when I first got here, I bumped into Mike Zock in the hallway in the science building, the A wing, I think it was. It was near the pendulum. I remember meeting you. And he comes up to me and he goes, "So, you have some history with the NSF?" And I said, "Yes, and National Science Foundation." And he says, "Have you ever heard of anyone getting a career grant who's at an undergraduate institution of this size?" A career grant is a major NSF grant of $400,000 to $500,000, which is given to promising young scientists from around the country. And so I had worked at the NSF for a couple of years, and I remember saying very clearly to Mike, well, it better be very special because there ain't many of those that go to undergraduate institutions. Lo and behold, Michael landed a career award. And truly, it's a spectacular achievement for an institution the size of Stevens Point. And so Mike we're just very honored to have Mike at the institution. But it turns out he's a local boy. And I didn't even know this until I started talking to him. He has a bachelor's degree in chemistry from UWSP. He has a PhD from the University of California, Irvine, and he was a postdoctoral fellow at UC Berkeley and was a Glenn Seaborg postdoc fellow at Argonne National Laboratory. And I'll mention Argonne again in a few minutes. He's an assistant professor of chemistry currently at UWSP. Before his PhD, he was an artist, goldsmith, and jewelry store owner in Monroe, Wisconsin. Where's Monroe, Wisconsin?
South of Madison.
South of Madison.
6 miles from the border.
Thank you. I'm a newbie to Wisconsin yet, so don't quite know all of my geography. His academic interest is encouraging students to believe they have a future in science and to consider careers in scientific research by emphasizing participation in undergraduate research projects. His research interests are in expanding the range of materials that can be deposited as nanowires using simple benchtop techniques such as electrodeposition, chemical vapor deposition, and using such nanostructures to develop chemical sensors or as building blocks for making smart materials. Smart materials. This is something that talks back to you, is that it?
Could. He
has numerous recognitions for his research, including a Micro Nano 25 award, an R&D 100 award, and has had his nanowire research featured on the covers of Science magazine. And is this the Science cover?
No.
This is not the Science.
That was in Science though.
What is this from?
So that was that won the International Visualization Challenge.
This is the International Digitization Challenge Image Award. This, you want to describe what this is?
So
in 30 seconds.
Okay, so the little dots that you see in the background are all bacteria. And the colorful part is a salt crystal. And essentially it's a war going on between the two. If the if the bacteria can't produce enough stuff to shut down the growth of the crystal, eventually the crystal is going to overgrow the bacteria.
30 seconds. He was named a WISIFS Innovation Scholar Award in 2008. And I thought this was an opportunity really to have Mike sort of kick off his talk, because this image was used as the cover, was on the cover of the National Science Foundation budgetary request to Congress this year. That's a pretty big deal, guys. A scientist from UWSP had his image on the cover of the National Science Foundation budgetary request to Congress. We're talking of billions of dollars request. So having said that, if I do this correctly here, this is also a cover which was on the ACS publication Applied Materials and Interfaces. Gives you a sense for Mike's status in the field. This is a picture taken of Mike down in, I'm going to turn the lights down just a little bit. Better? A few weeks back, Mike went back to Argonne National Laboratory down near Chicago and took some students with him. And do you want to tell them what this the name of this instrument is in 10 seconds?
So this is an ultra-high vacuum scanning tunneling microscope.
This is an ultra-high vacuum scanning tunneling microscope. Sure. You could have said anything and we would have nodded our heads. But suffice it to say, probably exorbitantly expensive instrument that you can't find on a normal college campus, especially like college campus our size. Mike has taken students down to Argonne at least the last 3 or 4 years, if I'm correct. And then finally, he also went down there to Argonne a few weeks back with my development officer, Julie Smith, who happens to be right here. And they suited up for the Clean Lab, which was at Argonne as well. It's my pleasure to present Mike Zock. The title of his talk is UWSP's Nanowires, High-Tech Cutting-Edge Research in an Unexpected Place. Welcome, Mike.
Thank you. So I'd like to thank everybody for coming out this evening. So the work that I have done that I'm going to show you today is essentially stuff that I started after I came to UWSP. So it's been all within the last 5.5 years. And essentially what it is we're taking and making very, very small wires. Just to give you kind of an idea of how small these are, you could take a bundle of 1 million of these wires, twist it together, and you're still not the thickness of a single strand of a spider's web. Yet they conduct electricity. We can take and make devices out of these, and we're really just starting to scratch the surface of this. So since this is so new and this is so different than other methods that are used to take and make the nanowires, really, it's kind of like planting a seed. I have really no idea where it's going to grow to, but you're seeing some of the very first images with this. So what we have here in the background is these are bismuth wires, and these are grown on an electrode, and I'll describe a little bit more in just a bit on how that's done. But bismuth wires are eventually going to be used in thermoelectric materials. So there are devices right now where you put electricity in, one side gets hot, the other one gets cold. In fact, it can actually be well below zero just in a small little device called a Peltier junction. Eventually, we're looking at putting those into there. So this is kind of a little bit of an outline of what I'm going to be talking about. The first part is really kind of a nanotechnology 101. And so nano is a prefix that means 1/1,000,000th, or 10 to the minus 9. So a nanometer is 1/1,000,000th of a meter. So meters this long, 1/1,000,000th of it is about that big. In fact, it's essentially if you put nano in front of any word, it just really means very, very small. So nanoparticle is a very small particle. Nanotube is a very small tube. Nanowire is a very small wire. So how small is small? And this is one of the things that's really kind of difficult for most people that are not in the sciences, and even people within the sciences, to really try and get a handle of how small we're really talking about. So let's start with something that's very familiar. So a meter, meter is about the length of a golf club. If we're going to take and measure, somebody's height, it's going to be somewhere usually between 1 and 2 meters. Millimeter, think of an ant. An ant is a good reference point for millimeter. Depending on the species of ant, it could be anywhere from just a couple of millimeters up to maybe about 5 millimeters for the largest ones, or the thickness of your fingernail. The thickness of your fingernail is about a millimeter. So now we get down 1,000 times less to a micrometer. And this is where it starts to really kind of get fuzzy as far as, so if you're to take a single hair, that's going to be about 60 micrometers. Diameter of spider web silk is going to be anywhere between 2 and 10 nanometers, or micrometers, depending on the species of spider, what type of silk it is. Diameter of red blood cell is somewhere around 3 microns in diameter. So, how many people have an idea of how big a red blood cell is? You see, we're already, so if you've looked at it under the microscope or tried to touch one with a little probe or something like that, it's really getting down there in size. So we're going to go even smaller yet. So a nanometer is 10 to the minus 9, or 1/1,000,000th of a meter. So a DNA strand is down around 2 nanometers. The cell wall for many types of cells is somewhere in the 20 to 30 nanometer range. We're really getting down there in size. So now if you think about the hair that's just 60 microns, and you think about how do you manipulate something like that. Here's a little experiment at home. Take a razor blade or a knife and try splitting a hair. A hair is 60,000 nanometers. Okay? So taking and making things very small from what we refer to as a top-down approach is very difficult. So taking larger objects, whittling them down, works to a certain point, but if you try this experiment of splitting a hair, you'll find out that it's very difficult. You need special tools to get much smaller, and just really isn't quite very easy. So, computer chips right now are mostly made with top-down approaches. We take and try to constrain the size of bulk materials. We remove certain parts in order to get things that are small. Okay, so if you take a look at state-of-the-art technology, so Intel's newest Pentium chips are produced in factories. Those factories are in excess of $9 billion just to take and make computer chips. So you can see that it's a really tough problem to try and get down to that size scale there. So how is that done? So how do we take and make things in that size scale? So the top-down approach that is most often used is called lithography. Litho meaning stone, graphy meaning writing in stone. We're not writing in stone, we're writing in different materials. So gold, platinum, semiconductor materials, and we use active patterning for this. So we pattern it with light. So to take and make a structure that's very small, we start off with a bare silicon wafer. So silicon is a very useful material. We know how to take and make it very flat. There's useful materials properties to silicon. But one of the things that we can do with it is we use it as a flat substrate to grow things off of. Onto that, we take and put a very thin layer of plastic. You know, a special type of plastic, we can drip it on, spin it out, and it ends up forming this very thin sheet on the top of that. And what's neat about this type of polymer is we can take and cover it with a pattern, and then we take and shine ultraviolet light down onto there. And what that's going to do is when we remove the pattern on there, it leaves a latent pattern behind. So we've changed the chemistry of that plastic. So now some of it we can take and put into a bath containing some solvents, and we remove some of that. So we can get things down fairly small with this. So the absolute limit of this is down somewhere around 45 nanometers. They're pressing 32 nanometers. Practical, boy, by the time you start getting down below a micron in size, this starts to get really expensive. In fact, you know, on a bench top or you know, with relatively simple tools, we're already having difficulty by the time we get down to 10 microns. So roughly on the order of the size of the spider web there. So if you want to get down even smaller yet, there's another technique called electron beam lithography. So in the back of the old style TVs, there's an electron gun, shoots electrons, and it can be controlled to move back and forth. And we can take and do the same thing in a special tool. Tool roughly costs about $1 million. And essentially we can take and turn that beam on and off and raster it back and forth and create a pattern. Think of that in some respects kind of like monks. The ancient monks transcribed books, and they would have to take and in order to take and create a finished page, they'd have to start off on the very first page writing the letter B followed by letter A followed by the letter B. They have to take and do things in order like that. Okay, same way. It's a relatively slow serial technique. Again, you take and remove the excess there to get just the pattern that you want. The limit of that is down around 10 nanometers. So we're getting pretty small with that. Practical, good question. It's not practical to take and make computer chips, and we use a lot of computer chips. It's practical really pretty much for research at this point. It's very expensive, it's a slow serial technique, and because of those it really is limited to just doing research. Okay, so now picture taking and building things up. If we take nature's smallest building blocks, individual atoms and ions, we can take and build things up. And in some respects, because we're starting with those smallest structures and building things up, we're working with nature. Instead of trying to take and confine something and remove something and using very expensive, high energy tools to confine the size of things, we can take and build things up. They're going to be small to start with because we're starting with very small building blocks. So now what connection do we have in central Wisconsin to the nanowires? So this is where my work is. So as I said, reference size, if we were talking about an ant, here's an ant that's a few millimeters. Now I didn't create the ant, but what we're really doing is we're trying to learn nature's secrets for making things very small.